Recording started.
Okay, at this point we are going to do a review of the DNA and protein unit, and this will cover some of the material that we need to know for the next test, but it's really important that you go over the review sheet that was handed out earlier, because that will cover a lot more information that's necessary for the test. So the first thing we're going to look at is DNA and RNA. And of course these are really important nucleic acids, very important compounds in our body. DNA, the name DNA comes from deoxyribonucleic acid. So you can see the D, the N, and the A. With RNA, it comes from ribonucleic acid. Okay, so the difference between these two names has to do with the different sugars that makes up each of them. And so deoxyribose is the 5-carbon sugar in DNA, and ribose is the 5-carbon sugar in RNA. Now both of these types of nucleic acids, DNA and RNA, are made up of nucleotides. So nucleotides are the subunits that make up either DNA or RNA. Alright, let's go to the next screen. So nucleotides are made up of 3 things: phosphates, sugar, and base. So those 3 things together make up a nucleotide. And now if you have just a phosphate and a sugar, that makes up not a nucleotide but a nucleoside. But once you add the base to the nucleoside, then you've got a nucleotide. Okay, so this is what's going to be joining together to make one strand of either DNA or RNA, a whole bunch of these. Now, the bases that make up DNA are known as nitrogenous bases. And the reason why that is because they all have nitrogen in them. So whether you're talking about adenine or thymine or whatever, they all have nitrogen in them. Okay, moving on to the next screen. So we see that in DNA we have adenine or A, guanine or G, cytosine or C, and thymine or T. And in RNA we have 3 of the same bases, adenine, guanine, and cytosine. But now, instead of thymine, RNA has uracil, or U. Now what are we looking at in the next diagram? Well, the next diagram is showing that we have a whole bunch of these nucleotide subunits, and when we join them together, we have one strand of DNA. So here's one strand of DNA made up of a number of nucleotides, and here's the other. And you can see that the bases are joined together. Okay, moving on to the next screen, we've got a few questions now that have to do with the material we were just looking at. So the first question, the repeating subunits of DNA are called. So what do you think those subunits are called? They would be called nucleotides, right? And again, remember the difference between a nucleotide and a nucleoside. Okay, going on to the next screen. In DNA, adenine, cytosine, thymine, and guanine are called. So what are they called? Well, we know they're called bases, but since they have nitrogen in them, they're called nitrogenous bases. Okay, screen number 6. And this question says, this base is never part of DNA. So which one is it? Well, it's uracil, or U. Okay, so again, DNA has thymine, or T, but in RNA, instead, we see uracil, or U. Okay, next question. In DNA, adenine pairs with what? And in RNA, adenine pairs with what is it? Okay, so let's find out. Well, once again, we have A matching with T with DNA, and with RNA, we have A matching with U. So these are the 4 bases and the 2 pairs that we see with DNA, and here they are with RNA. And again, it's just A and T or A and U that makes the difference. Okay, now what is the main sugar in DNA? That's our next question. Well, it has to do with the D, right? What is it? It is deoxyribose, and again with RNA, it's ribose. Okay, now at this point we're going to look at an important, very important process, which is DNA replication. So this is the process in which organisms use to make more copies or duplicate copies of their own DNA. Now the way a DNA molecule replicates itself is in two steps. First of all, it unzips or separates its two strands, and then it completes each separated strand by creating a new strand which has bases that match with the bases on the unzipped strand. Okay, well let's look at how this works. So here we see a DNA molecule which has unzipped itself into two separate strands, and you can see that these strands have now been duplicated already. So in the beginning it was just a single strand here, and now it's been duplicated. Each one of them has. Alright, so you can see the two strands have been unzipped, and at the bottom you can see the separated strands. And basically what's happened is we've had to find bases that pair with each of those bases on the unzipped strands. Alright, now what are we looking at here? There's a whole bunch of stuff on this diagram, and I just want you to focus on a few things. Just focus on the DNA polymerase and what it's doing, and the DNA ligase and what it's doing. These are both special types of enzymes that are involved in DNA replication. So, as the diagram shows, DNA polymerase, that's this one here, has the job of adding nucleotides or units of P, S, and B to the unzipped strand, the blue strand on the right. Okay? So we're going to take this strand and we're going to find some nucleotides that match. So we're going to find bases that are going to match with the bases on this unzipped strand. So here's a better picture of what's going on. So you can see here, this is the unzipped strand, this bottom one. It's kind of like the blue one we had. And we found some nucleotides, and we added them one at a time. And then we're going to have a double-stranded DNA again, because we're doing DNA replication. Okay, so that's DNA polymerase. Let's have a look at the other enzyme. Okay, so what is DNA ligase doing during DNA replication? So that's going to be this one, focusing on this part right here. Well, you can see that a number of nucleotides have been joined to the unzipped strand on the right, and this creates new DNA called Ozarky fragments. Okay? So these are these fragments here. They've been created, and but there's just a whole bunch of them right now. So what does DNA ligase do? It joins all these fragments together. Okay, so that's what you can see with the yellow here. It suggests that we're joining these fragments together. Okay. All right, so a few questions to test what we've just gone over. Now, the process in which the DNA molecule separates or unzips to produce two identical replicas is called, well, that's DNA replication, right? DNA replication takes a DNA molecule and produces an identical one from it. Okay, now this question says a series of enzymes called blank fuse free nucleotides in the complementary chain of DNA. So we're going to be fusing together or joining free nucleotides together with the chain of DNA. Okay. Now the enzyme which joins nucleotides to the complementary strand of DNA, in other words, the unzipped strand of DNA, is DNA polymerase. Okay, so it's joining nucleotides to the unzipped strand of DNA. This question says, a blank is a biological glue that permits one section of DNA to be fused to another. And that was the other important enzyme we looked at. So the enzyme which joins the new DNA fragments being made together is DNA ligase. So DNA ligase is like a glue that joins DNA fragments together. Those were the Ozarki fragments. Okay, the next question says, if one strand of DNA had the following nitrogenous or nitrogen-based sequence, okay, so here's a DNA sequence, the other strand will have what sequence? Okay, so this is one strand of DNA. What's the other strand of DNA going to look like? Well, we need to take the fact that the following matching of base pairs, A and T, and G and C, are used to create the DNA sequence. So here we have A and T, we've got G and C. Okay, so we just look at the C and we know that G is going to pair with it. And we see here, we have G, C is going to pair with it. So we create this new DNA sequence. All right, so that's our answer. Now, do you remember where DNA replicates? Where does it, or DNA replication occurs? Where does it occur in the cell? And when does it occur? Well, it occurs in the nucleus of a cell, DNA replication does, and it happens before cell division. Now, why would it happen before cell division? It makes sense that before cells divide, they would need to double the amount of DNA that they have. So that's why this is when DNA replication takes place. All right, great. So we've reviewed what DNA replication is all about. DNA doubles or replicates itself before cell division takes place. But do you remember those two other important processes, what they're all about? Transcription and translation? We shall find out on the next screen. All right, so DNA is often used to produce proteins. You know, the body has a lot of important proteins, whether we're talking about hormones or enzymes, those are all proteins. Actually, some hormones are based on steroids. But there are a lot of different important building blocks in the body that are made out of proteins. So the overall process is taking DNA to produce RNA, and then RNA is used to produce proteins. So the step where we go from DNA to RNA, that's called transcription. And the step where we go from RNA to produce proteins, that's called translation. And so, okay, so during transcription, DNA is used to produce an equivalent copy of RNA. All right. So transcription and translation. So here's what it looks like. All right, so at the top you can see this RNA molecule, which is called messenger RNA. So that was produced from DNA. That was copied, that's an equivalent copy from DNA. And as you can see, a protein molecule is being assembled by joining together a number of amino acids. So these are all amino acids, and when they're joined together, they're going to produce a protein. Okay, so transfer RNA, that's this guy right here, has 3 base sequences. And this is known as a triplet. And these are going to match, okay, this 3 base sequence is going to match with a triplet on messenger RNA. So we're going to find another 3 base sequence, and it's going to match together with this one. And then once you get the match happening, all of the amino acids are going to join to form a protein. So it's really important to know the overall process of what's going on with transcription, translation. We've already seen DNA replication earlier. Okay, so here's a question that's going to kind of apply what we were just looking at. If we started with the DNA strand below, what would the amino acids be in the protein that would be created? So we're starting with DNA. We know we're going to have to go to RNA and then eventually produce a protein from it. So on the diploma exam, we would be given a chart that looks like this one. and that would help us answer the question. So what we need to do is look at the same process that occurs in organisms, where we take a DNA strand and see what our RNA molecule is produced, and then we're going to look at each triplet or 3-base sequence in the RNA molecule. Remember, that's with the transfer RNA. So an example of one is going to be AUU. And from that, we're going to see on the chart which amino acid is going to be produced from that triplet code. For instance, here's a triplet code of GCG, and we see that the amino acid alanine is going to be produced from that. Okay, so that's the basics of what we're going to be doing. Let's go to the next screen. All right, so what we do then is we take the DNA molecule we were given in the question, and we take what we know about matching, and we do the matching up. So we know that with RNA, instead of having A match with T, we're going to have A match with U, and again here, and then G matching with C all the way along. So we produce our messenger RNA molecule. That's the first thing we do in this question. And what are we going to do next? Well, next what we're going to do is we look at each triplet or 3-base sequence in the RNA, and we're going to see which amino acid these triplets are going to match with. So we need to take this messenger RNA sequence and divide it up into 3s to produce triplet codes. Okay, so here's what I've done. I've divided it up into 3. So we've got our triplet codes. Okay, now notice, this is an important thing to note as well. Transcription, so again, when we're going from DNA to RNA, that occurs in the nucleus of a cell. The same thing with DNA replication, that also occurs in the nucleus of a cell. Whereas translation occurs in the cytoplasm of a cell. Okay, so that's where all that, it's not empty space, but it's mainly liquid that's in the cell. That's where the proteins are being produced. All right, so let's have a look at a little bit more at how we use this chart. So we took the original DNA, we got an RNA molecule, we divided it up into these triplet codes. We're going to look at the first one. We've got UUC, and we see that it matches with phenylalanine. So we look on here for UUC, and there you go, phenylalanine. So that's the first amino acid that's going to be produced from our first triplet code. If we look at the next one, next one's going to be proline. CCG is going to be producing proline. So if we look on here, CCG.
Goodbye.
All right, produces protein. The last one, GAA, that's going to produce glutamate. Okay, so that's GAA. And sorry, I'm just trying to find it here on the screen. GAA. Should be glutamate. I can't seem to see it at the moment, but it should be there. Okay, glutamate. There we go. Sorry. GAA, glutamate. Okay, now the last one's important to pay attention to. The last code we have, UAG, it doesn't match with any of the amino acids here. It matches with something called a stop sequence. Okay, so UAG is right here, produces a stop sequence, so that would tell the protein that it needs to stop being produced from the amino acids. So in other words, we've only got 3 amino acids. So even though we had 4 triplet codes, we've only got 3 amino acids. And so these 3 together are going to produce the protein that we have. So usually a protein is many, many amino acids. So usually when you only have 3 of them, you would call it a polypeptide instead of a protein. But sometimes those two terms, polypeptides and proteins, are used interchangeably. Okay, so that would be our answer for this question. So again, we looked at the original DNA strand, we looked at which RNA molecule was produced from it, we divided it up into triplet codes, and from those codes, we looked on the chart, we found the amino acids, we joined them together to produce our final product. Okay, next question. This one says, use the following information to answer the next question. So what we see here is we see some original DNA. It's being used to produce RNA, and then it's being used to produce proteins. So we're looking at these two processes, and it says state the location in the cell where process 1 occurs and the location where process 2 occurs. Okay, so we're going from DNA to RNA. Where does that occur? Again, that's transcription. And RNA to proteins, translation, where does that occur? Well, again, transcription as well as DNA replication, they both occur in the nucleus, whereas translation, where we produce proteins, occurs in cytoplasm. Good stuff. And I believe we are on the last screen, or maybe the second last. Okay, so we're going to look at some important enzymes, some other enzymes, other than the ones we've looked at so far. These are called restriction enzymes, and their job is a little different. Rather than building up DNA, restriction enzymes are used to break up DNA into smaller pieces of DNA, or genes. These enzymes are found in many organisms. Now remember, whether you're looking at genetic engineering, sometimes it's called gene splicing, that involves taking the DNA of an organism and adding more DNA to it. And sometimes the new DNA is coming from another organism. The new DNA is produced, that is produced after genetic engineering, is known as recombinant DNA. It's kind of like you're recombining. Okay, so this is the next question we're looking at, and it says state which enzyme would be used for each of the three processes above, which involve a portion of human DNA being added to the DNA of a bacteria. Now, since bacteria grow quickly, this allows human genes to be produced more quickly. So what we're doing then is we're breaking down DNA here, human DNA, and we're also breaking up the DNA of the bacteria, and then we're adding the human DNA into the bacteria. So here the human DNA is added into the bacteria. So we've got recombinant DNA, new DNA. And so basically what that's allowing you to do, since humans don't reproduce very quickly, you're able to take a gene from a human and put it into bacteria, and then because bacteria do reproduce quickly, you can reproduce that or produce that DNA, the human DNA, very quickly using the bacteria's DNA. Okay, so what we need to do is we need to state which enzyme is going to be used for each of these processes. Okay, so the first is breaking down the bacteria DNA. The second process is breaking down the human DNA. And the third one is joining together fragments of DNA. So which types of enzymes are we going to use? Well, for both the first two processes where we're going to break down DNA, we're going to use restriction enzymes because that's their job. And for the third process where we're going to join DNA fragments together, that's going to be a ligase enzyme. And we saw that earlier when we were looking at the process of DNA replication. We're joining together those Azarki fragments. All right, so I believe that's it for now. And again, I really want to encourage you to go over the review sheet that we had to prepare for the next test. If you have any questions at all based on that sheet, please let me know. Have a great day. God bless.